Yes, thanks for the introduction, Keith. Last week you heard from my PhD student, Antonia Musso, who talked about the attack dynamics work that we're doing in collaboration with Alan Carroll. And today I'm going to be talking about more of our work looking at factors that influence flight. And this work that I'm going to be discussing today, a large portion of it was done in collaboration with Nadir El-Bilgan and his lab members. So this is Kelsey Jones, and as I mentioned previously, this is the what I'll be presenting today is the work of Kelsey, who graduated with a Master's of Science from my lab recently. Okay, so I know that everyone in this audience is aware that mountain pine beetle is an important ecological and economic component of our pine forests, as Keith introduced earlier. It can feed and complete development in almost all species in the genus Pinus. And it's such an important economic component because of its ability to overcome the defenses of healthy trees to allow for host colonization, which is mediated by pheromone-mediated mass attack. So mountain pine beetle goes through different population states, which are characterized by different densities of beetle on the landscape. And this is what this graph is shown. So on the y-axis, we have the mountain pine beetle population density on the landscape. So during the incipient epidemic and epidemic population phases, the mountain pine beetle attacks and kills healthy pine trees on the landscape. Most of the life stages of the mountain pine beetle occur under the bark. Eggs are laid under the bark in the late summer, and larvae develop feeding on phloem in larval galleries and overwinter in late larval instars. Pupation occurs in the subcortical environment, and emergent teneral adults will continue to feed until emergence in mid to late summer, when adults fly to seek new hosts for brood production. So this dispersal part of the life cycle that occurs after the adults emerge from the natal host is obligatory when beetles are in the epidemic population phases, because the natal host will already be dead and unsuitable for brood production. So a lot of the work in our lab focuses on this dispersal component. This is an important part of the mountain pine beetle ecology to study, because in Alberta, we are dealing with this recent range expansion from outbreak populations of mountain pine beetle in British Columbia that were present in the early 2000s. As part of this range expansion across the Rocky Mountains into Alberta, the beetle has encountered a new host, jack pine. And I'm sure you've seen these maps many times. One of the important things, of course, is that jack pine extends across Canada's boreal forest. So part of understanding the likelihood of mountain pine beetle eastward range expansion is to better understand beetle dispersal and the factors that affect dispersal by flight. And then, of course, after dispersal comes the host colonization process. And so we're also interested to understand how flight impacts host colonization. So the mountain pine beetle exhibits 3 distinct patterns of population spread as a result of flight. The first 2 patterns, shown on the left-hand side here, occur within a forest stand. So spot growth occurs when emerging beetles attack a tree near the natal host, while spot proliferation is when beetles fly past suitable hosts before selection of a host further away, but still within the same stand. These short distance dispersal flights that result in these two types of population spread are extremely variable and can range from only a few meters to some tens of kilometers. The third type of dispersal is long distance dispersal, where the beetle movement is aided by the wind. And long distance dispersal events can result in movement over hundreds of kilometers. In my lab at the University of Alberta, we've been studying flight in mountain pine beetle for quite a while now. And we have the results of some of these findings are listed here. So some of the main findings include that bigger beetles fly for further distances and for a longer duration. We've also shown that lipids are the main metabolites used to power flight, but that other sources, including carbohydrates and proteins, are also important. We've shown that abiotic conditions, including wind, photoperiod, and temperature, influence both the timing and duration of flight. And we've been trying to uncover why flight capacity of mountain pine beetle is so extremely variable, as this variation can't be completely explained by any of these factors that we have studied to date. So just as a little bit of background to some of the studies we'll be conducting that I'll be discussing today, I just want to introduce some of the important message-bearing chemicals that are important for the beetles in their behavior on the landscape. So beetles use these message-bearing chemicals, which are referred to as semiochemicals, for a variety of purposes, but they have to respond to them to orient in their environment, which facilitates a variety of things, but most importantly, host colonization. So they are sensitive to and respond to volatile signals emitted by host trees, non-host trees, such as trembling aspen, and to chemical cues produced by other mountain pine beetles or conspecifics. So these message-bearing semiochemicals can be attractive or deterrent to mountain pine beetle. Attractive semiochemicals include some host volatiles that aid in orientation to stands of host trees, as well as close range assessment of host tree suitability. So some examples of these include the tree-produced compounds such as alpha-pinene, beta-philandrine, and myrcene. Beetle-produced attractants include things like aggregation pheromones that promote orientation to a focal tree and facilitate that mass attack process that I introduced earlier. These aggregation pheromones include the female-produced aggregation pheromone transverbenol and low concentrations of the male-produced aggregation pheromone exobrevicommin. Other semiochemicals are considered to be deterrent, and some deterrent semiochemicals include volatile organic compounds released from non-host trees, such as nonanol and benzyl alcohol, released by trembling aspen. The beetles themselves also produce deterrents, such as anti-aggregation pheromones, like frontalin and verbenone, and these signals indicate to conspecifics that the tree is full and they shouldn't attempt to colonize. And even it's been shown that even high concentrations of the male-produced aggregation pheromone exobrevicommin deter beetle orientation. So because we know that mountain pine beetle exhibits this extreme variation in flight behavior that results in differences in dispersal among individuals, we wanted to examine factors that influence this variability and determine if the variability is important for subsequent host colonization behaviors. So in Kelsey's work, our objectives were to determine if semiochemicals influence mountain pine beetle flight capacity. And this should be clarified that this isn't how semiochemicals influence orientation of the beetles, but rather how exposure to these semiochemicals impacts the beetle's willingness to fly and flight distance and duration of these flight bouts. And then we also conducted experiments to determine if flight variation influences subsequent host colonization behaviors, including pheromone production of colonizing beetles. So I'll just go over some of the main methods that we used in all experiments before getting into the individual experiments themselves. We used beetles in all of our experiments that were obtained from infested lodgepole pine trees from 2 sites in the expanded range in Alberta. The tree bolts containing the beetles were held in the lab in emergence containers, and the adult beetles were collected daily and separated by sex, and then held for subsequent use in the flight bioassays. So flight assays were conducted using computer-linked flight mills. And the way that these work is that the beetle is attached via a tether to the mill, and as it flies around, a magnetic sensor is tripped on the flight mill, and that is recorded to the computer that is attached to the flight mill. And so once the flight is over, we can then use this information to understand, to measure flight distance, flight duration, the number of flights, the flight speed, etc. So the first section of Kelsey's thesis examined the effect of semiochemical exposure on flight distance of female mountain pine beetles. She conducted a number of experiments, but they can be sort of categorized into 2 groupings. One was to determine if semiochemical exposure before flight would influence subsequent flight capacity of the beetles. And then secondly, to determine if semiochemical exposure during flight influences flight capacity. So in the first experiment, we tested the effect of pre-exposure to female-produced aggregation pheromone, transverbenol, on flight capacity of female beetles. This was compared to control beetles that experienced the same conditions but exposure to simply clean air. So this is the setup that Kelsey used. She positioned the beetles in these exposure chambers where they were exposed to transverbenol or clean air for a 3-hour period. And this exposure included the semiochemical or the clean air delivered in a stream of purified humidified air. The exposure lasted for 3 hours, and following exposure, beetles were then positioned on the computer-linked flight mills and were allowed to fly for 23 hours. The total flight distance and duration was measured for beetles that experienced either clean air or the transverbanal treated air before their flight. So from this experiment, Kelsey was able to show that beetles exposed to the female aggregation pheromone prior to flight flew longer distances than beetles that received no prior exposure to transverbanal. The second experiment that Kelsey did was to assess how beetle flight is affected by exposure to host and non-host volatiles during flight. So we chose to look at the impact of volatiles emitted from the phloem of the historic host of mountain pine beetle, lodgepole pine, and the novel host, jack pine, as well as the non-host that is present in the environment where mountain pine beetle are, and that's trembling aspen. So exposure to these compounds during flight was compared to beetles that were flown in clean air. Kelsey collected phloem discs of each species from the respective tree species and then transported the phloem discs back to the lab in liquid nitrogen, where they were held at -80 until used in bioassays. So this is how the setup looked. So in our flight mill room, the flight mills were grouped into groups of 4 mills, and those mills were enclosed with an inert material. And then they were treated with one of 4 treatments. Either they did not have phloem discs positioned in the arena, so shown by the X's in the blue box, and that's the clean air treatment, or they had phloem discs from one of the tree species. So either lodgepole pine, shown in red, jack pine, shown in yellow, or trembling aspen phloem, shown in green. So the treatment position in the flight mill room was randomized between enclosures with each bioassay. So the position within the flight mill room varied depending on the replicate. And between replicates, the enclosures were sort of dismantled. The material used to enclose the flight mills was replaced, and the mills themselves were all washed to prevent chemical contamination between replicates. So from the results of this experiment, we can see that the presence of tree volatiles during flight altered the distance that beetles flew on the mills. The main difference was that beetles exposed to the trembling aspen volatiles, shown by the green bar, flew for shorter distances than the beetles in clean air. Flight in the presence of both the historic and novel pine hosts was intermediate between that achieved in clean air and in aspen treated air, but was not significantly different from the clean air control. So to summarize the findings from this part of Kelsey's research, we showed that exposure to the female produced aggregation pheromone transverbanal increased the flight distance of female mountain pine beetles when they were exposed prior to flight. When females were exposed to tree volatiles during flight, Kelsey's work showed that pine host volatiles present don't really influence flight distance, but exposure to aspen volatiles decreases flight distance of beetles. So these findings could have several implications for mountain pine beetle movement in its expanded range. Increased flight distances after exposure to transverbenol may help to promote dispersal away from occupied areas. And this might be particularly important in jack pine, as higher levels of transverbenol are produced from beetles that attack jack pine. Host volatiles don't really have a significant effect on dispersal capacity, as the exposure to these pine volatiles during flight didn't affect the flight behavior. But a patchy environment with large areas of trembling aspen may decrease dispersal success of mountain pine beetle, as aspen volatiles resulted in less flight by beetles in this study, and they've also been shown to disrupt oriented flight to attractive semiochemicals in previous work. So the next part of Kelsey's work focused on the influence of flight itself on the subsequent host colonization and pheromone production in mountain pine beetle. So we know from previous studies that mountain pine beetle condition influences subsequent host colonization. So beetles with a low body condition are less likely to enter hosts, and of the beetles that do enter the hosts, those with the worst body condition enter the host first. So this is the experimental setup for examining the effect of flight behavior on subsequent host colonization and pheromone production. So male and female beetles were exposed to a flight treatment where they were flown on the computer-linked flight mills for 23 hours, just as we used in the first part of Kelsey's work. Now, a subset of the beetles were not given the opportunity to fly, and these beetles served as a control. After the flight treatment was applied, we could assess the total flight distance and duration that the flown beetles conducted. So following the application of this flight treatment, both flown and control beetles were introduced into uninfested lodgepole pine bolts. The beetle activity and that's shown in the middle panel of this slide. The beetle activity was monitored for 72 hours to determine if and when the females entered the host. For the beetle pairs that entered the host bolts, female and male pheromone production was assessed by aeration of beetles that established in the host material. So these volatiles released by the beetles were captured on PORPAC Q and then extracted for analysis by GC mass spec. So our results on the effect of flight on host colonization support the earlier findings on the impact of beetle condition on host colonization. So we found that beetles that did not expend energy in flight were more likely to colonize tree bolts than beetles that experienced the flight treatment. So the control beetles were 14% more likely to colonize bolts compared to the flown beetles. Further, this effect seems to be driven by energy loss, as weight lost during the bioassay dictates the propensity to enter the host, with beetles that lost less weight being more likely to enter the host. And those, of course, were the control beetles that didn't have the opportunity to fly. We also found that of the beetles that flew prior to host entry, and those are shown by the green bars in this graph, the females that lost the most weight during flight were more desperate and entered the host more quickly than those beetles that lost less weight during flight. So the next part of the study measured the effects of flight on pheromone production. The female released aggregation pheromone transverbenol is produced from the monoterpene precursor alpha-pinene and released by the female upon feeding on the newly colonized host. The male produced aggregation pheromone exobrevicolamin is produced de novo in the fat body and initially released after leaving the natal host tree. So what Kelsey found was that female transverbenol production actually increases with distance flown by females. This increased pheromone production with flight appears to be driven by energy use, as transverbenol production increases with weight loss during treatment. In contrast, male aggregation pheromone production decreases with increasing flight distance of male beetles. And exobrevicolamin production decreases with energy use, measured by weight lost during the treatment. So, in summary for this part of her work, Kelsey showed that host colonization post-flight is driven by energy use, which supports these previous findings on beetle condition. So beetles that lost the most weight during flight did not enter hosts. But of those that did enter, the beetles that lost the most weight entered the fastest. Flight also impacted pheromone production of both female and male mountain pine beetles, but in the opposite direction. Females that flew further produced more transverbenol, whereas males that flew further produced less exobrevicolamin. The implications for this work suggest that there is a trade-off between flight and colonization that will select for shorter flight distance. However, the increased transverbenol production with flight distance will select for longer flights and may assist in beetle movement through patchy environments like it might encounter with eastern expansion through the boreal forest. Selection on the opposite sides of this dispersal spectrum will likely maintain the flight variation that we see as an evolutionary stable strategy. Finally, I'd like to acknowledge all the various people and funding bodies that assisted with this work, and particularly Kelsey's work was funded mostly by the TriaNet project, with many contributors to that funding pool. I'd be happy to take any questions that you might have, and I also leave my contact information here. Thanks for listening.